Good day. In today's lecture, we will be discussing about membrane transport of small molecules and the electrical properties of membranes. Now, in the previous part or in the previous lecture, we have discussed about the structure of the lipid bilayer and of the different proteins embedded in the lipid bilayer. Now, because we have the lipid bilayer, it actually separates the interior of the cell to the exterior. And because of this separation, we have a difference in the concentration of ions. So if you remember, ions cannot pass through the membrane. So you have here a difference, the concentration gradient of these different charged particles. So for example, we have here our sodium ion, which is which has a larger amount of sodium ions outside in the extracellular space rather than the interior of the cell, which is about 5 to 15 millimolars and versus 145 outside. And then the opposite is for the potassium. There are much more potassium ions inside than it is outside. So for the magnesium, it's just a difference of 2 to 4 folds difference, but for the calcium ions, you have a really large amount of, or rather, compared to the other ions, you have a very, very little amount of calcium ions inside the cell, but but we have compared to the outside, which is a large amount. So inside versus the outside. And then for the pH, we relatively maintain a pH of 7.2, so relatively neutral. So the interior and exterior are very similar. Although in some cases, such as inside the mitochondria or in the chloroplast, where the proton gradient is important, there is a difference in the proton ion concentration. But generally, they are somewhat similar. And then for the anions, we have large amounts of chloride ions outside than it is inside. Now, from as a review from the previous part, so we have the lipid bilayer. So the lipid bilayer is made up of phospholipids. So you have a polar heads and a nonpolar tails. And the nonpolar tails, because they are hydrophobic, tend to be situated on the interior of the bilayer. And because they are nonpolar, polar and charged particles cannot pass through them. So here is the permeability. So if it's a hydrophobic molecule, whether it's a small molecule such as oxygen and carbon dioxide and nitrogen, or even large molecules, as long as you are hydrophobic, such as the steroids, steroid hormones, and other basically hydrophobic hormones, they just easily permeates, they easily passes through this membrane because of the hydrophobicity of the interior. For the small uncharged polar molecules, so they are polar, but they are small. So, such as water, urea, glycerol, and ammonia, they are generally impermeable, but even a small amount can still pass through. So they are permeable in a pure lipid, in the pure bilipid layer. They can pass through, but only small amount. So there's a large proportion that is being rejected from the bilayer. So it's they can filter in for a small percentage can pass through. For larger but uncharged polar molecules, even smaller because they are larger, they have much more difficulty in passing through the bilayer, unlike the smaller uncharged but polar molecules. And then for the ions, even though they are very small, such as the proton ion, basically this is just a naked proton, that's the H plus. Your hydronium ion, even if they are very, very small, they absolutely cannot pass through because the hydrophobic interior of the lipid bilayer is repelling them. So you have this permeability. And so the factors affecting the permeability of the bilayer, we can say that the first factor is, of course, hydrophobicity. Because regardless whether you're big or small, as long as you're hydrophobic, you can generally pass through. And then the next factor is the charges. For the polar molecules, you have small, which can pass through, although a smaller percentage can pass through, majority still being repelled, but still they can pass through in a greater capacity than the larger polar molecules. And lastly, the charge. If you are charged, you cannot pass through the lipid bilayer. So this permeability, we can actually have a way to calculate this permeability. So the permeability coefficient for the passage of various molecules through your synthetic lipid bilayers. So when we say permeability, this pertains to the rate of flow of the solute across the bilayer. And this is directly proportional to the difference in the concentration of your membrane. So basically, the permeability, we can calculate how fast the certain solute passes through the membrane by multiplying our concentration difference and the permeability coefficient. So the permeability coefficient is an inherent property of the solute. It determines its whether it will pass through or not. For example, hydrophobic molecules from our previous example here, they pass through, so they have a high permeability. Small uncharged molecules are mostly repelled, so they have a lower permeability. Permeability coefficients. And then for the larger one, much smaller permeability coefficient. And then for the ions, almost negligible permeability coefficient. And we use this permeability coefficient to determine how fast the solute is passing through the membrane, because although they can pass through, what actually drives them when we are talking about passive diffusion here, what actually drives them is the concentration gradient. So we need to have a concentration difference multiplied by the permeability coefficient. This will give us an idea how fast the solute crosses the bilayer. So for the solutes that cannot pass through the bilayer itself, we have transport proteins that allow them to cross the transport proteins. So these are special membrane transport proteins. Their jobs are usually to transfer the polar and charged molecules, because again, they are the ones that are being repelled by the hydrophobic bilayer. And so we have here, they require special assistance for them to cross the bilayer. So there are two major classes of membrane transport proteins. You have your transporters, which some books or some resources call them carriers, or some others call them also permeases, because they help in the solutes to permeate the bilayer. And then another class is the channels. So what's the difference between them? So this is your transporter and this is your channel. So let's look first at transporters. So transporters, or the permeases or the carriers, so they bind specific solute to be transported and then undergoes a series of conformational changes. So for example here, this is a glucose transporter. So you have here a glucose. A glucose, remember, is a large polar but uncharged molecule. So they can pass through but only a small amount. So they require, and remember, glucose is an important molecule for the cell because it's actually the source of energy. So the cell must be able to intake a lot of glucose. And how will it be able to do that? It's through the glucose transporter. So for the glucose transporter, your glucose must first bind to the transporter. And once bound, it will undergo a conformational change. So as you can see here, at first, the opening is to the outside. So this is the outside of the cell, this is the interior. The opening is from the outside going to the, and then close to the interior. And then once the glucose molecule is bound, you have now, it closes to the outside and opens on the inside. And then afterwards, the glucose will then dissociate. Once the glucose becomes unbound, it will revert back to its original conformation, which is open to the outside. And that is how your glucose transporter can carry glucose from the outside to the inside. So from here, you can see that transporters or carriers, they are quite specific to what they are transporting, because the material must first bind to them, and they move, or their mode of action is similar to an enzyme. So you have binding, conformational changes, and then the reaction basically here is the transportation of the solute from the outside to the inside, and then reverting back to its initial conformation. So it's very similar to how an enzyme reacts. So for the case of the channels, as its name suggests, they are open on both sides. So unlike the carriers, which are only open, mostly open on one side, and then undergo conformational change to actually transport the material. For channels, They are open on both sides already. And unlike the transporters, their interaction with the solute they are transporting is much more weaker. They do not have to specifically bind their solute. So because basically they just allow them to pass through. So they form continuous pores that extends across the lipid bilayer. So example here is your potassium channel. So here you can see there's a continuous channel here, or a continuous tunnel, somewhat like a tunnel, that allows your potassium ions, the green ones, to pass through. So because they do not bind the solute, they are able to transport ions. Usually it's the ions, and actually ions and water. They also have water for water channels. They are able to transport these solutes, these materials, much faster than the than your transporters, because they do not have to undergo conformational changes every time they transport a batch of solutes. So they just allow them to pass through, just like a highway. So you just pass through, pass through, pass through. So they are able to transport it at much faster rate. And they have the ability to become much more selective. For example, here, your potassium ion is very selective. It transports potassium, but it doesn't transport sodium. Now, if you remember from your chemistry, so potassium and sodium are on the same family. And they are both positively charged, +1 ions. And although the difference is that potassium ions is larger than your sodium ions, your channel, and both of them have the same charge, but your channel can actually distinguish between sodium and potassium, and it only allows potassium to pass through but not sodium. So that's one characteristics of your channel. They are highly selective, and that's because of the selectivity filter. Usually they are found at the opening of the channel, and in this case this is on one side of the, usually the exterior side of your channel. So channels are mostly passive transporters, and when we say passive, so they just follow along the concentration gradient. So we do not have to exert ATP to transport materials against the concentration gradient, and that's actually makes sense because channels really just binds to the solute weakly, and they just allow it to pass through. So they generally follow the concentration gradient. But the way to regulate the channels is by allowing them to open and close. So just like breaches, for example. So if you do not want motorists to pass through a certain from one island to another, for example, you do not want them to pass through, you just close the bridge. So basically, this is the way to for the cell to control what passes through, the what goes in, what comes out from their what goes on, what comes in from the interior to the exterior to the interior. Now, the examples of these channels, we have your acetylcholine, or rather acetylcholine receptor here given. This is a ligand-gated channel. So the way to regulate them is through the binding of a ligand. For example, this acetylcholine receptor, we call it acetylcholine receptor, but in reality, it's actually a channel. It's a sodium ion channel. But we call it acetylcholine receptor because it requires acetylcholine to open the channel, and it's basically the receptor for acetylcholine. So acetylcholine is a type of a neurotransmitter. Usually we find them in the motor neurons and in the neuromuscular junctions. They are responsible for the movement of basically voluntary movement mostly, and not only, although they are not only limited to the voluntary movement, but also in the neurons in the brain. But anyway, you have your acetylcholine that must first bind. So the general conformation of your channel is closed. Acetylcholine will bind to the channel and then it will open and allow the sodium ion to pass through. So take note, although it's called acetylcholine receptor, it doesn't allow acetylcholine to pass through. What passes through this channel is actually the sodium ions. And then another one is the voltage-gated channel. You usually find them along the axons of neurons. They are most of their role is in the propagation of the action potential. Now, for the voltage-gated channel, they are sensitive to the difference, to the charge difference in the membrane, and they open or close depending on the depending on the charges, the charge differences. When they sense a depolarization event, so they open and allows your, well, if it's a sodium channel, it allows sodium to pass through. If it's a potassium channel, it allows potassium to pass through. So you have your gating that basically inactivates your channel. We will discuss more on voltage gated channel because they, unlike the ligand gated channel, the voltage gated channel actually has three modes. So we will discuss more on this one later. So we have types of membrane transport and membrane potential. So we have, of course, the passive transport, which does not really require much energy. They follow the concentration gradient, or in the case of ions, we have an electrochemical gradient because the charges of the ions also matter. So the passive transport, they occur spontaneously. So basically diffusion. So simple diffusion. So we have simple diffusion where your material just passes through, usually for hydrophobic ones, and then channel mediated or transporter mediated for a passive transport. And then we have the active transport, which requires metabolic an input of your metabolic energy. So they are usually mediated by the transporters because transporters can actually forcibly move materials across the membrane, whether the material wanted to or not. They have no choice but to move, unlike the channels where it's mostly passive. The movement is passive following the concentration gradient. So we have, for the active transport, the molecules there are the transporters, or sometimes we call them pumps. So as I mentioned earlier, aside from the concentration gradient, we also have the electrochemical gradient. Actually, it's the electrical gradient. So the electrochemical gradient combines the membrane potential and your concentration gradient of your solute. So when you talk about membrane potential, it's the difference between the charges on the exterior, on one side of the membrane, versus the other side of the membrane. So here you have a difference in the membrane potential. So in here, we have just simply a concentration gradient. So looking at the simple concentration gradient, for a passive diffusion, we have the push is just from the lower to the high, from higher to lower concentration. Now, when we factor in the electrical potential, so the charge differences across the membrane, we have a slightly different outlook now on how ions, especially ions, travel across your membrane. Because for ions, they are charged molecules, so whether they when they cross the membrane, they will affect not only the concentration gradient but also the electrical gradient. So here are two modes on how the electrical and the chemical gradients can be rather two scenarios on the electrical and chemical gradient. So for here, we have scenario 1. So here, we have a large concentration. When we look at simply concentration, larger concentration outside, smaller concentration inside. So of course, according to the chemical gradient, just the concentration gradient, the movement is from the outside to the inside. And then looking at the charges, we have an excess of positive charge on one side, the outside, and a negative charge on the inside. Of course, positive, same charges repel, different charges attract. So the movement is again from the outside to the inside because these are positive ions. So for positive ions, you want to go to the negative potential. So we have here the electrical gradient and the concentration gradient moving with each other. So we have a thicker arrow, so there is a larger push for the movement from the outside to the inside. So that's scenario 1. For scenario 2, we have here again the concentration gradient, large concentration outside, small concentration inside. Of course, according to the concentration gradient, they want to enter the cell. But when you look at the electrical gradient, we have a positive charge outside, a negative charge inside. So the tendency is for the negative charges, because they are negatively charged ions, the ions wanted to stay outside according to the electrical potential. But for the concentration potential, they want to go inside. So the two gradients are working against one another. And as you can see, the movement still follows the chemical gradient, but it's being prevented by your electrical gradient. So I'm not saying here that the chemical gradient always win in every scenario, but rather these two are basically their effects are additive. It's just that there's a much greater chemical, the push for the chemical gradient, or rather the concentration gradient, is larger than the electrical gradient. So you have a movement across the channel, but the slimmer arrow indicates that it's smaller, or the push is much smaller. So there only a little amount can passes through in a passive diffusion mode. Unlike this one, there's a larger push to go inside. This one, because the electrical gradient is working against it, so you have a smaller initiative to cross the membrane. So basically, these are the two types of membrane transport potential. Now, in the next part of this lecture, we will be discussing more in depth about the transporters and the active membrane transport.